Thanks, and thanks very much for the opportunity to be here. We're not going to talk about funding because everything that was in my bio changed, shall we say, and that's all we're going to say about funding. Yeah, thanks everybody for coming. I'm going to try and give a sort of a whip through of where we are in terms of big concepts in HIV vaccine design. Those of you know me know that I always focus on antibodies, so you'll see it's an antibody-heavy talk as usual. There's also quite a lot of data in some of the slides which I'm not going to discuss at all. I want to get through the concepts rather than get into the details, all of which are published. But the first challenge is going to be whether or not I can break the podium. And I wanted to start with this slide, which is a reminder of how variable HIV is. I get, and I'm sure all of you get the question often about, if it was so easy for us to come up with a vaccine as a community for SARS-CoV-2, why is it so difficult for HIV? And this slide really summarizes how vastly different the two pathogens are. On the left-hand side, each one of those colored ticks is a mutation, and you can see SARS-CoV-2 is fairly mutated, as we know, which is why those vaccines became problematic quite soon. But HIV is just like orders of magnitude more variable. Each one of those colored dots is a mutation, and the amount of variation in one person living with HIV is equivalent to global diversity for flu. So we are dealing with a very different beast here in terms of vaccine design. One of the reasons that we have hope that an HIV vaccine could trigger broadly neutralizing antibodies, which are the kinds of antibodies that would be able to recognize that huge diversity of viruses that we know exists, is that some people do naturally, after many years of infection, and that's the key, after many years of infection, they do make those broadly neutralizing antibodies. And so while those antibodies don't benefit the people in whom they develop, the hope is that if we can get those antibodies and trigger them by vaccination beforehand, then that would be a different outcome. And people like my team have spent decades, and literally decades, studying one or two or three individuals who happen to make those broadly neutralizing antibodies and really defining the arms race very carefully and understanding exactly how those antibodies developed in people who did develop those antibodies. And understanding that pathway, which has, as you can see, been the basis of many wonderful collaborations and many wonderful PhD and master's projects, means we have a roadmap now in terms of understanding what we're trying to do. We've studied people who are living with HIV, we understand how it happened in them, and now we can try to replicate that process without the virus by vaccination. And that's what is now the basis of a phrase you'll hear often in the field, which is rational vaccine design strategies. And when I was showing my slides to my 16-year-old kid, he said, "Huh? So what have you guys been doing for the last 20 years if what you've been doing up to now has not been rational?" Which is a fair point, I thought. What we mean here by rational vaccine design is an even more engineered version of what we were trying to do. We used historically kind of more empiric ways of trying to design vaccines, but now we're going back to that roadmap that we have learned from people who are living with HIV who develop broadly neutralizing antibodies, and we're copying it literally at the molecular level. So it's a much more thoughtful, careful, orchestrated program of trying to drive B cells along the right pathway, even though it is a bit of a silly name. So I said I wanted to cover some of the key concepts that I think are changing the field. I'm not going to try and cover everything in 15 minutes, and I know Betty speaks after me and will cover some more of these concepts, but I really wanted to touch on four main approaches that I think may change the HIV vaccine landscape, have already. The first is something called germline targeting, which you will have heard a lot about in the field. The whole field seems obsessed with germline targeting at the moment, and I'll try to explain what that is. But three other concepts which I think are worthy of focusing on is the idea of actually immunizing people living with HIV. So this is testing vaccines in people who have already become infected, and I'll explain the logic there. Pediatric trials of vaccines, and then slow delivery immunization. And the reason I'm focusing on those four concepts is I think they have the potential to shift the HIV vaccine landscape, but also other variable pathogens. So germline targeting is based on our understanding now that those people who made broadly neutralizing antibodies do so because very, very, very rare, ultra-rare precursors in their bodies were stimulated. So it's like looking for hen's teeth in those individuals. If we were to apply germline targeting to this audience, each one of you would have between 1 and 3 of the correct B cells. Only 1 in 3 of the millions of B cells that you have in your body. And what germline targeting tries to do is to engineer a vaccine that specifically goes in and finds those 1, 2, or 3 B cells that are in your bodies and stimulates them to become more. And the idea is that by finding those hen's teeth precursors and setting them off along the right pathway, you can drive them towards broadly neutralizing antibodies. This work was, it's unbelievably elegant immunological discovery work, published not so recently, 2023 in Science. But I just wanted to make the point that these germline targeting vaccines have shown great promise in the sense that they can activate those 1 to 3 precursors. But actually we have a very long way to go before these antibodies become a true vaccine. We are literally taking the first step in this strategy. And so when people say we have an HIV vaccine in the form of a germline targeting vaccine, I would argue that that's not yet a reality, and we need to be very careful, I think, in how we describe the immunology. So we've taken the first step in germline targeting towards triggering the right vaccines, but have a very long way to go. And I think that's clear from the way in which we measure success in these germline targeting vaccines. Betty's sick of hearing me talk about this because in any vaccine where we talk about trying to measure these antibodies, it's one of the key roadblocks is that the only way you can tell whether these vaccines have even started along the right pathway is by doing this crazy amount of next generation sequencing and trying to understand whether you've triggered exactly the right antibody. The old-fashioned now ways of measuring vaccine success used to be by simple binding assays, by ELISAs and by neutralization assays. These vaccines, the germline targeting vaccines, are so far back in that process that doesn't work. And you can only tell by sequencing millions and millions of B cells whether your vaccine has worked. And I think that's an indication of how far we still have to go in this field. I also think that, that there's a challenge in expanding germline triggering to other classes. In people who develop broadly neutralizing antibodies, they develop a whole swarm of antibodies. And this approach of germline targeting is based on the fact that many people develop the same kinds of antibodies to one single site of vulnerability on the HIV envelope, and that's shown in this figure down here. We all do it in the same way, which means that we can engineer vaccines to trigger that. But almost no other antibody in our repertoire has that ability. So again, just to make the point that germline targeting really looks for a crazy rare subset of antibodies and tries to stimulate them to produce enough to be able to protect people. So it is a difficult ask, is the point of all of this. Okay, I want to move on to 3 other concepts and then take a little bit of a step back and reflect on where we are. The second concept I mentioned is testing vaccines in children. The reason we think that this might be viable is if all of us were living with HIV and I were to screen this whole audience for broadly neutralizing antibodies, maybe 10 of you would have those kinds of antibodies, maybe. But if we were all 4, and I know I sometimes behave like that, but if we were all 4 years old and I were to screen all of you for antibodies, more than half of you would have those types of antibodies. There is something intrinsically different about the pediatric immune system, which in the context of infection means that those kids develop much higher levels of broadly neutralizing antibodies, somewhere between 50 and 80%, and I'm showing on this slide studies from multiple sites across Africa. So the rationale for this is that if kids, when infected with HIV, are able to make broadly neutralizing antibodies, perhaps they would also respond better to a vaccine regimen that attempted to trigger broadly neutralizing antibodies. And so this is one of the major concepts in the field, is whether we can leverage on that difference in the pediatric immune system to test vaccines in kids. And if that were to be successful, then perhaps we could leverage on those years before sexual adulthood to be able to improve vaccine efficacy. And although this has a long way to go in terms of what we know about it, there are several studies now in young humans and in young macaques that do support the idea that this might work. The other key change in the field is this understanding that maybe giving 2 shots is not the best way to get your immune system to respond to vaccines. This is work that was really based on what we know from people living with HIV, where, as I said, it takes years and years and years to get to the point where they have these broadly neutralizing antibodies. And this is work largely out of the Scripps Research Institute that showed that if you take exactly the same amount of vaccine that you would normally give in one shot, and you give it in 7 small shots over 7 days, you get much better responses than if you gave the same amount of vaccine as one bolus. This is a sort of a sequential slow delivery system. In NHPs, they used osmotic pumps to do this, but as I say, in humans, what we're doing is giving lots and lots of little shots, which to many of you in the audience who are medics must seem like a nightmare, and probably is a nightmare, but you know, there are ways we can get around that. For example, slow delivery release systems. Putting a plaster on somebody that slowly administers the same amount of vaccine would probably have the same effect as these osmotic pumps in NHPs. The first data that's coming out of humans here looks interesting too, and so I think combining all of these ideas might work. And then the last concept that I really want to focus on before I tell you of yet more challenges is the idea of polishing immunogens rapidly in people living with HIV. This is based on the idea that we've been way too slow as a field in testing vaccine concepts, and it takes years and years and years, as you all know, to get to the point of being able to say, no, actually this vaccine is rubbish. If we had a better way of testing 20 to 30 vaccines, we all have ideas. If we had a better way of testing them faster, then that could advance the field. But testing those vaccines in people who are not infected with HIV, as I said, takes years and years and years. But people who have been infected with HIV and have subsequently been put on antiretrovirals could be considered to be perfectly primed. Their bodies have seen every version of HIV, every version of the HIV envelope that is included in these vaccines. And the argument here, which we describe in this commentary, is that if those people, still under ARVs, still themselves fine, if those people do not respond to a vaccine, then an HIV-naive individual is extremely unlikely to respond to a vaccine. So it's a way of testing how good our polishing, boosting vaccines are. And I think these kinds of trials, more of which are starting, can really shift the vaccine field. So those are the 4 concepts, that I also just want to remind you of what we learned quickly from the AMP trial, because I think it's been a sobering couple of years. The AMP trial was the trial that tested one of these broadly neutralizing antibodies in thousands of people to ask whether a broadly neutralizing antibody could in fact prevent infection, and it can, which was a very happy outcome for those of us who've spent our whole years trying to understand how to trigger these antibodies. But what was sobering was that the bar was much higher than we thought. So we needed much more antibody than we had previously thought in order to prevent infection. So now take that statement and go back to what I was saying about having to next generation sequence those germline targeting vaccines, and you can see how far away we still are. In addition, by sequencing and studying the viruses that did infect people in the placebo arms of the AMP trial, we learned that circulating viruses are becoming more resistant. As the virus is evolving at a population level, the viruses are becoming more resistant to certain BNAVs. So that lifts the bar even higher. Thirdly, we had always thought that the transmission bottleneck was extremely stringent. So what that means is that in a person who's exposed to HIV, generally our understanding was that one virus, one virion, maybe 2, got across in most cases. But the work from the AMP trial, largely from Jim Mullins and Carolyn Williamson, shows that actually, even in the absence of an STI, many more viruses are getting across. We just weren't counting them because we weren't sequencing deep enough. And it turns out that nearly half of infections are caused by more than one virus particle, which also lifts the bar yet further. So we need enough antibody to prevent 1, 2, 3, maybe 4 different viral lineages from getting across to people. And then, and this is work we started, Betty and Linda Gale and others within the Brilliant Consortium, starting to try to understand the gaps again across the African continent. And this slide shows you the huge gaps in our understanding of the viruses that are circulating in Africa. Combined with the fact that many of the viruses that we are looking at are changing, becoming more resistant, I think this really highlights the need for us to go out into our communities and understand which viruses are circulating and whether they would be sensitive to the kinds of antibodies we're trying to trigger. There's also a notion in the last 20 seconds that mRNA platforms are going to change the world and this is going to solve all our problems for HIV vaccine design. I don't think that's the case at all. mRNA platforms may enable us to go faster. A lot of that depends on how effectively we can manufacture mRNA, and it turns out that's hard. And as you all know, there are also issues around safety with urticaria. But even without those two issues, the mRNA platform doesn't change the intrinsic challenges that are with HIV that I showed you right on my first slide about how variable the virus is. So I think it'll enable us to test lots of concepts a lot faster, but it's not the silver bullet. And then, of course, I've, as usual, spoken largely about antibody responses because I think that's actually where much of the cool immunology is happening at the moment in the HIV vaccine field. But it's clear that we would need T-cell vaccines as well, both on their own merits and also to be able to drive enhanced maturation of B-cell responses. And there's this elegant study from NHP's really showed this, and obviously many more people working on those kinds of concepts. So I guess trying to summarize all of this, we need much more antibody than we realized to prevent HIV, and very unhelpfully, the viruses are becoming more resistant. The transmission bottleneck is less stringent than we thought. And those strategies that we do have a long way to go. But they are on the right track, and testing our boosting vaccines in populations such as people living with HIV in pediatric populations, I think would provide faster and essential insights for acceleration of vaccine discovery. And with that, I will thank all our funders and my team and hand over to my colleague, Betty. Yeah, thanks.